Translate that mRNA.
The lead role is mRNA. After RNA processing, it has a 5' cap and poly A tail with 10 codons. Next up is the supporting cast. It is the 9 tRNAs with its amino acids and 1 stop codon, which is the release factor. In our project, we use the same tRNA to bind to the same codon, which is AGG. The second lead is ribosome, or rRNA. It contains large subunit and small subunit. A site means amino acyl tRNA binding site, where tRNA enters. The P site means peptidyl tRNA binding site, where polypeptide attaches and grows. Above the P site is the exit tunnel. The E site is the exit site, where tRNA exit. The amino acyl tRNA synthetase catalyzes the production of tRNA with amino acid.
The specific amino acid, which in this case is MET, binds to the amino acyl tRNA synthetase. Then ATP phosphorylates and adds an AMP to the amino acid. An amino acid is covalently bonded to the tRNA by the AMP, and then BAM! tRNA with a specific amino acid is covalently bonded! Wow!
First, the small subunit binds to the mRNA. Then a large subunit comes to complete the complex and start translation. For the very first codon on the mRNA, which is AUG, the codon must be placed under the P site, so the tRNA with an anticodon, which is UCA, enters the ribosome and into the P site to bind by complementary bases and remains there until another tRNA enters the A site and binds to its complementary bases. The amino acid currently in the P site is catalyzed by the large subunit to create a peptide bond with the amino acid in the A site. Then the tRNA will move one codon and the tRNA in the A site will move in the P site. GTP was used to move the mRNA. When the tRNA moves to the E site, it will then exit out to the cytosol where it will be recharged by the amino acyl tRNA synthetase.
Finally, when the mRNA reaches to the poly A tail, it encounters a stop codon, which in this case is UAG. The release factor has an anticodon, AUC, which is complementary to the stop codon. When this occurs, everything disassembles and the protein detaches. Then, the protein folds based on its R group properties and ta-da! A protein is born.
Yeah.